Okay, thank you, Adam. Now here comes the trash talk. Let's talk about garbage that is generated in every household all the time in the form of, unnecessary and broken things. These are collected by the garbage trucks, which transport their cargo, ideally to the recycling centers, where all this trash is sorted, transformed, and used to manufacture new products. And if this system breaks down, then garbage piles up on the streets, as we have seen a couple of years ago in Naples, Italy, and that's not good. And our cells also have a garbage recycling system, which is based on lysosomes. These organelles can be considered the stomach or the recycling center of the cell. So lysosomes degrade all kinds of biological macromolecules into building blocks, and then these are recycled back to the cytoplasm for reuse, for making new molecules or to generate ATP energy. Now, cargo can reach lysosomes in multiple ways. Cells can take up things from the environment, also from their own plasma membrane, and form small bubbles called endocytic vesicles. And these will fuse with lysosomes to deliver their cargo. But cells can also cannibalize themselves. They can engulf portions of their own material, cytoplasm, including the protein-making factories, ribosomes, or the powerhouses of the cells, mitochondria, to capture these into vesicles that will also fuse with lysosomes, and then this recycling process can happen. Now, lysosomal degradation, unfortunately, is not always perfect. Especially during aging, lysosomes tend to accumulate more and more undegraded cargo that is known as lipofuscin or the H pigment. It's evident if you look at the skin of an old person, these H pigments are all over the skin cells. But not just the skin cells, in every cell, in neurons, in muscles, and of course it's not good if garbage is piling up in our cells. This can be even worse when there is a large-scale accumulation of undegraded cargo, as is seen in many diseases, such as Alzheimer, which is a good example. Here in these electron microscopy pictures, you can see this is a so-called dystrophic neurite. A nerve process that neurons use to communicate with each other. And here you can see all these black structures. These are all endocytic and autophagic cargo that is not being degraded properly. From a healthy brain, if you look at the same cross-section, same magnification, this is a normal neurite. By now we know that autophagic degradation, lysosomes, are very important for human health, and lots of diseases are associated with defects in autophagic lysosomal degradation. We know this thanks to the pioneering researchers. The first one of them was Yoshinori Osumi, who discovered the first genes required for autophagy in yeast. And since the homologs of these genes are present in all eukaryotes, we could actually study the importance of autophagy by disrupting these genes. And since the biomedical importance of autophagy became evident, he got the Nobel Prize 5 years ago. Now, we in our lab use human cultured cells, as most molecular biology labs do all over the world. But we also use Drosophila, the fruit fly. Now you can ask the question, why the fly? Well, the fly is a very useful animal model. it has all the organs and the developmental programs that we have. It has a brain, it can learn, it can sleep, it can get drunk, and it becomes addicted to alcohol. In our lab, we use actually it as a brain injury and recovery model. It has stem cells to renew its body. It's aging. Of course, it's short-lived, only 2 months. So aging experiments are really fast in flies. And the programs that determine how the body develops are so similar that if you mess up the same signaling pathway that leads to a formation of an extra finger in affected patients, the same manipulation will give an extra bit of the wing in flies. So it's very quick and cheap to work with it. In 2 months, we can generate the entire population of London and just keep it on the shelf. And because of these advantages, fly researchers already got 10 Nobel Prizes for their discoveries. And actually, the majority of genes whose mutations are causing disease in humans have their homologs in flies. And if you disrupt these genes, you can also induce Parkinson's disease, diabetes, or cancer in flies. So this is a cancer mutation effect. This is a wild-type fruit fly. Look how the head and the thorax becomes abnormal due to the excess cell divisions caused by a cancer mutation. And you can use flies to develop medicines. I will tell you an example later on. So one of the studies I would like to mention today, we did together with human geneticists who found in a Turkish family 2 boys, both of whom inherited homozygous mutations for one of the autophagy genes. So this mutation caused just 1 amino acid change in the corresponding protein. And these boys suffered from neurodegeneration, movement disorders, known as ataxia, and mental retardation. And we used gene editing to introduce the same mutation into flies. And we got neurodegeneration, movement disorders, so this is the wild-type flies, this is the mutant fly, and a very short lifespan. So autophagy is really important, and its defects cause disease. So one of the biggest projects that we undertook in our lab was to search for new autophagy genes. So for that, we disrupted every gene in the fly, one by one, in selected cells in the larval fat tissue. Here you can see 2 green cells. And these 2 green cells lack 1 gene called syntaxin 17. And as a result, you can see that the pattern of autophagic structures seen by magenta here on the left in the composite image. But here you can see the grayscale image channel of just the autophagic structures. You can see that in these 2 cells lacking syntaxin 17, all these puncta are much smaller and fainter, indicating that there is a problem with autophagy. Compared to these control neighboring cells, which have bigger and brighter punctae. Now, this gene encodes for a snare protein, and snare proteins are known to be important for the fusion of 2 vesicles. These are depicted here. In this case, it's autophagic vesicles fusing with lysosomes, with the help of syntaxin 17 in control cells, which is not happening in these 2 mutant cells. Now, we found a number of snare protein coding genes. We used biochemistry to show that these really form a complex. And when we knocked out disrupted syntaxin 17 in Drosophila, we did, again, these climbing assays. And you can see that the wild-type control flies climb normally, whereas the mutants, well, they don't do much, and they also have very short lifespan. So it's really affecting autophagy and causing ataxia. Another common test that we use in microscopy is to look at the localization of proteins in cells. So here is the fat tissue again. In blue, you can see the cell nuclei. And this boxed area is enlarged here. In green, we are looking at a protein that we are interested in called RAP2, and in red, we mark lysosomes. And if you look at the separate green and red channels, you can see that here are the lysosomes, and the green protein called RAP2 is forming very nice rings in these optical sections. So this protein is present on the surface of lysosomes. And we disrupted it, we found that it's required for all kinds of vesicle fusions, including endocytosis and autophagy, going to the lysosome. Now, what I've been telling you is kind of like basic research, so we try to understand what's going on in these cases with the hope that it will be useful later on. But we also do some applied research, which is targeted at let's say, immediate problems to improve the human condition. A good example is the success story of mRNA vaccines that was based on discoveries done earlier by Katalin Karikó and others. For example, that RNA induces inflammation in cells, and you have to modify them so to avoid this inflammation response. So this applied research that we are involved in is actually a drug development process, but it's not de novo drug discovery, because that means you need to invest a lot of time and so much money that only pharma giants can mostly afford that, and the success rate is very low. What you can do is you can take known drugs and you can try to find some of them, perhaps for a new disease that they can cure. It's called drug repurposing. And how we use that in the lab is if we have human patients with a known genomic lesions that cause their disease, we can generate a personalized Drosophila model using gene editing. And then take these sick flies and try thousands of known drugs and see if any of these can actually help the sick flies. And if we found such molecules, we can go back and try on patients. And remember, these are known and already clinically used drugs. So the whole process is much faster, a couple of years. So we are part of a consortium involving multiple groups where we are doing such drug screens, trying to cure selected rare human diseases, for example, using survival or just this locomotor assay, and see whether we can find a cure for a rare disease. And with that, I will conclude my talk to thank my team, the funding agencies, and I'd like to mention that in my lab there are currently 2 junior mentors, Tamás Maruzs and Áron Szabó, and we have 2, well, actually right now 1 academic student, my namesake, Gábor Juhász, and another student is joining us in January. So thank you for your attention. Here's a cartoon to depict how autophagy is going on in cells while you are, coming up with your questions.
Thank you, Gábor, for this really exciting presentation. And I would like to ask the audience whether there are any questions. Please be active if there are any thoughts. While others are thinking, I would have a generic question. I mean, I'm not sure everyone is aware that basically you mentioned the replacement of uridine by pseudouridine. And actually, it is because RNA in its original form would stimulate toll-like receptors, at least 3 types of them, which were initially discovered in Drosophila, and this became a huge success story for inflammation research. So along this line, I would like to ask you, which are the next big hits you would imagine to influence translational research from the Drosophila field and more closely from the autophagy field? So can you maybe tell another example which could become similar to the success story of toll-like receptors?
Well, of course, it's difficult to predict. If you could predict, you could always publish in the best journals what will happen. But what I really like nowadays, and this is going on big time in the United States, is that they sequence cancer patients, I mean the cancer genome of patients who are not responding well to therapy. And now with this new gene editing CRISPR system, they can generate the fly model in 2 months and then just run a panel of drugs through these model flies. And actually, it's fast enough so that the patient is still alive and they have the results. And they are already using this in the clinic, the fly model. So I think, I'm hoping that we will see more and more of these really translational use of fruit flies.
Thank you. Again, I would like to ask if there are any questions, please.
Sorry, I cannot hear you.
Speak up.
Well, it's different, right? I think in general, of course, if it's closer to human, it's better. Actually, mini pig is, I think, for now, is very popular in pharmaceutical companies. But flies are much faster to work with. And actually, a lot of the Nobel Prizes go to yeast researchers. As you have seen, the autophagy Nobel Prize went to yeast researchers. Because that is the fastest and the simplest eukaryotic model. So all the basic cell biology, how cells divide, how autophagy is going on, this is discovered in yeast, and they do it. But of course, you cannot model neurodegeneration or movement disorders in yeast. For that, you need, well, say, flies. So it depends on what you want to use it for. I wouldn't say that there is a best model.
Thank you, Gábor.